I've decided to do Lab 5 a little bit differently. I've made guide videos for you before, and I know some of you have struggled a little bit with questions on the labs. Some of the questions will be answered explicitly in this video. Others are going to make you think and stretch a little bit. If you're stuck on any of them, please feel free to send me an email. Let's make an appointment, go over them. The idea is to stretch you a little bit, make you work for some of them and make some conclusions, putting things together. Okay, I'm going to start with a little bit more detail about opsin molecules. We've talked about how opsin molecules are these G protein coupled receptors, which is a large family of cell surface receptors. So they receive a signal on one side and then transmit that signal internally into the cell. Now, ancestrally, these signals were chemical. So some chemical would bind to the extracellular portion of the membrane protein, and that would cause a conformational change in the molecule. And then that conformational change would result in some sort of a change inside that would then, and it's in reaction to the G protein that's coupled to the inside. That G protein would then be released, and then there'd be a cascade that would allow for a signal to be transmitted. And so opsins come from that ancestor of a chemical signal, but they have been modified somewhat. Now, with proteins, there are many different ways that we can view them. And these are all have their purpose and serve different functions. So what I've got here is a diagram of different ways to look at the protein. So let's start here on the left. They've got the C terminus on one side and then looking at the opposite side with the N terminus. So this would be like looking at it from the extracellular portion. This would be like looking at it from the intracellular portion. And this is a space-filling molecule. So if you could magnify them, and we have microscopes that will do that, but usually not at physiological conditions. And so it might look different under what we could see with a microscope. But if we could, this is kind of what you would see. And they've color-coded this to look at different areas that are highly conserved versus ones that are a little bit more flexible and changeable residues. And so we'll look at that here in a little bit. So I don't care that you worry so much about all the color coding here. You may be more familiar with the ribbon model here. The ribbons represent secondary structure in the proteins. So these are not really completely accurate, but are representational of a helix, in this case, of what occurs in the molecule. Now for the opsin, and we've got three different views of this ribbon model here. This is the one from the side. And so here's the C terminus, here's the N terminus, and then the 7 transmembrane domains. Okay? So this is, again, to give you some idea of the structure, but be a little bit more diagrammatic, and you can see a little bit more than you can with just the space-filling model here. And so each of the 7 transmembrane domains is color-coded. You can see each of them, but it's still kind of busy and a little bit cluttered. So unless you knew a lot about the molecule already, you wouldn't be able to completely understand this diagram. And then finally, on the left here, we have the simplest, and it's very diagrammatic. It's not meant to be accurate at all. It's a two-dimensional version where it's a very three-dimensional molecule, but it is maybe the most as far as being able to convey information. And so I've got a version of this one also in the lab. And I've got a version of the ribbon model here. So let's take a look at the lab, and then we'll address the two questions here in the lab, at least in part, maybe even a little bit completely. Okay, so this is what you'll see in your lab. So I've got a version here of this two-dimensional diagram that's labeled, and I chose this one. It's a little simplistic, but I think that is helpful in some ways. Okay, so on the bottom, we've got the extracellular portion down here. That's the N terminus. And the C terminus is within the cytoplasm. So this would be outside the cell down here. This would be inside the cell up here. And so notice that we've got this inside portion, this C terminus, that then begins what is called, and this is just by convention, but this is transmembrane domain number 7. So by convention, we start numbering them over here where the N terminus is. The reason for that convention is this is where translation begins. So translation goes from N-terminus to the C-terminus. And so we've got this part that is outside the cell, and then a internal portion that's between the membrane. This would be the phospholipid membrane. And then we have these different areas. We have these loops. This is a cytoplasmic loop, and then this would be a extracellular loop. So these are little parts of the protein that are poking outside of the cellular membrane on various sides. So we've got 3 cytoplasmic loops, and we have 3 extracellular loops, and then we have the C-terminus and the N-terminus, and then all these 7 transmembrane domains. And each of them has a particular function. Some have more important function than others. Of course, the primary function of the transmembrane domains is to keep it anchored into the membrane in its proper position. Now there's a secondary part here that I'm going to look at with this ribbon model here in a little bit that is also an important function for opsins in the transmembrane domains. And so historically, ancestrally, the extracellular portions, N-terminus and perhaps the extracellular loops, although they're a little bit small, either individually or as a group, would receive the extracellular signal, the chemical signal, if you will. And then the C-terminus and the cytoplasmic loops, again, and I don't have a good idea about this. There may be some work that's done on this, but I honestly don't know the answer to this. But either individually or all together, because they're internally, they could then activate the G-coupled protein and help to transmit a signal. And often these, when we have a protein, these domains that are all physically close together, they work together to do whatever their function is. All right, so that is a simplistic diagram of opsin. Now, last thing I want to talk about this, we're going back to this ribbon model. So we see the 7 transmembrane domains color-coded here. The C terminal up here, the N-terminus. The last point that I want to make out is opsin is a little bit different than the ancestors, because ancestors would receive the signal with the external portions, right? These internal portions are not available to a signal that's a chemical signal externally. However, opsins have changed a little bit. The way that the signal is received, the opsin molecule, is there is a retinal molecule. Retinal, it's an organic molecule. This is a diagram. If you've taken organic chemistry, these will make sense to you, but it's not really important. But the idea is that when the correct wavelength of light interacts with these retinal molecules, it changes the conformation. So this is a light-sensitive molecule. And that change in conformation is the way that a light signal is then transduced into a chemical structure in the opsin molecule, or actually in the retinal molecule associated with the opsin molecule. They're not covalently bonded together, but they are associated. And then that then can result in the signal. So it's a little bit of a shift. So the signal is now received in the molecule itself down in the membrane. And so because membranes are thin and with enough light can penetrate quite deep down into cellular membranes. And these, of course, are stacked and arranged in a very complex manner in the retina of whatever organism we're looking at. So retinal, it was named because it's very common and at high concentrations in the retina. So a couple of questions here then. I just want to give you a little bit of background, maybe even answer these for you as we move forward. So based on the location of the different opsin domains, which of the domains are candidates for interacting with the G protein? So remember, the G protein is inside the cell. Okay, so that should give you a pretty good idea about which of these candidates are potential objects that we would look at, parts of the cell that we would look at for interacting with the G protein. Now, the last thing that I want to do is review this term. I think it's somewhat you're probably familiar with, but if not, this is an important concept. So a domain is a region of a protein that has a specific function. And so proteins can often be broken down by what their job is in the final amino acid structure. And proteins will often interact together. So for instance, we can see that all 7 of these transmembrane domains fold together and interact to make the transmembrane region of the protein. So that's their function. And so they need to have certain characteristics that allow them to be stable inside that membrane and serve their function. And mutations and changes that modify that function greatly, unless it is an advantage, which is unlikely, there's going to be some amount of stability and some amount of range that these domains could have in the amino acid sequences. And by the way, I think you maybe already realize this, but each of these individual circles represents an individual amino acid in the final protein. So if you didn't realize that, that's what they are. And they're not labeled in this one. In the one I showed you earlier, each one was labeled with a letter, so you could tell what amino acid it was. Okay, so that's what a domain is. And then one last thing about domains, if you've taken the molecular evolution class, you already know this. But in many cases, not always, but many times, domains correspond to exons in the gene. And so that allows for some shuffling, some mutations that can move domains around. And so it's good to think of these domains as kind of functional units of a protein. Often, maybe like a motorcycle, right, has different functional units. You have the engine, you have the wheels, you have a transmission, you have brakes, and each of those has a very specific job and is a separate portion of the motorcycle. And then sometimes different parts come together to act, like the brakes interact with the wheels so that you can stop it. So in the same way, these proteins have different parts that serve an individual function. Sometimes those individual parts interact together for a larger function. So think of that as a domain, and now you know what domains are. So the last question for this first part. Why might some protein domains be more conserved than others? Now, I'm not going to answer this explicitly, but let's talk quickly about, and we're actually going to look at this in section 3 of the lab, how we might estimate conservation. What do I mean by conservation? So for instance, if I look at a single Drosophila opsin, I can, even if I know the structure very, very well and can identify every single domain, I don't know anything about conservation of those domains because I only have one instance. It would be looking at like one language in the human society and saying, hey, how much do languages evolve? How conserved is this dialect or is this language? And if you only have one person speaking that language, you only have one instance. So you need to gather multiple samples. So we would maybe gather samples of that language from near, gather far away, look at closely related languages, and then we could begin to talk about the evolution, right? The changes that had occurred in that language. So the same thing is true of proteins. In order to estimate conservation of different domains, I would need to gather multiple samples of proteins that were all part of the same family, all closely related and shared a common ancestor. And so these could be orthologous copies. So if this is maybe Rhodopsin 1 in Drosophila, I could then look at Rhodopsin 1 in a mosquito, and then maybe another fly, and then maybe go beyond another insects or even other arthropods, or maybe even farther beyond all the organisms that had copies of that gene. And I would line them up and then look at where differences occurred. I could do that at the DNA level and also at the amino acid level. And we're going to use a piece of software in section 3 of this lab. You're going to learn a little bit about it, and you'll need to download it, and that will allow us to do this. So if you're not sure about this answer yet, think about it. You may already have some really good ideas. If not, then come back to it after we look at conservation of opsin in section 3. For the second part of Lab 5, I'm going to introduce you to a website. You can Google Ncbi and you'll find it. If you'd like to follow along, that's great. If not, you can look at it later. We'll be interacting with Ncbi a little bit. We'll be doing it through a piece of software that I'll introduce in the next section also, but I'd like to just talk about it a little bit. Even though we're not going to be using it a ton, it's good for, well, I think any biologist to know what it is. So Ncbi is run by the US government through the NIH. It is a data set, a repository for pretty much all of the genetic data that has ever been generated. Almost every journal and almost all institutions require that when you publish a study that uses DNA data, when you sequence a genome, anything like that, you have to deposit that in a publicly available place. And that place is maintained by your tax dollars. So Google Ncbi, it's the National Center for Biotechnology Information. When you go, it's a massive site. There's a ton of information here. and lots of tools also. We are really only going to scratch the surface, but we'll talk about things that you can do here. Okay, so you have a number of different databases. Let's start with two of these databases that I would like to introduce to you. You can look at genomes, you can look at proteins, but we're going to look at first taxonomy. So if you're interested in studying a specific organism, you can find the sequences for that organism by searching for their name. So I'm going to put in Drosophila melanogaster, the common fruit fly that's used in the laboratory. And then you'll come up with a variety of results. I did very, very specific, so I only came up with one single species. There it is, fruit fly, a species of fly. I'm going to click on that link, and then it gives me a lot of information about different projects, different ways that I can interact with data for Drosophila. Okay. Now, one of the things over here, it says, look, we have raw nucleotide sequences, we have protein sequences, we have different structural elements, we have a complete genome for that one. So you can and we have even population sets. Since Drosophila is a model organism, there's a ton of different information for Drosophila. If I click on this link here for nucleotides, it says there are 1,337,000 different nucleotide sequences available. If I click on that, it's going to list them beginning 20 at a time. So I could look through all of them individually and scan through them. Very inefficient. But in addition, one of the things that it does is it brings up a taxa ID here. That's a unique identifier for this species. So if I leave that there and then search for something else, now notice it defaulted to nucleotide once I did that because I clicked on the link for nucleotides. So now I'm searching for nucleotides in the Drosophila species, and let's look for opsins in Drosophila. Okay, so I'm just going to put a keyword here. And so basically what this means is search for nucleotide sequences in this species that have a keyword opsin. And when I do this, I'm going to now narrow down my 1 million sequences. I only have 69 sequences, much better. And again, because Drosophila is a model organism, oftentimes they're entire chromosomes. So this is a chromosome that has an opsin on it. Here's a gene called RH5. Here's one and gene names are a little weird. We've talked about that. Here's RH2. And so I'm just going to pick one of these. Here's the first one, Drosophila melanogaster opsin gene. Just kind of a very general title. Now, when you click on one of these, you're going to get a lot of information about where this sequence came from, right? So you have a location. You have an accession number, which is a unique number assigned to every single sequence. You have the species, you have keywords here, right? That's how we were able to find it. You have a little bit about the taxonomy of the organism. And then you have information about the publication or the study from which this sequence came. Okay, so kind of cool. This is going back probably a little bit older, 1985, which is very early for DNA sequence, but Drosophila is a model organism. And that's probably why it only says opsin. At the time, they were unsure how many opsins there were, and so they said, here's an opsin. And so later, it probably has a more specific name that was given to it. We could sort through some of these informations. And notice it has information about introns in this one. And for some sequences, this information is not available. But it has lots and lots of information. Okay. Now, one of the things that I would like to show you is looking at different formats for how we present DNA information. So at the very bottom, we've got, so this is the non-coding sequence in between the gene, which you can expand so you can see it, right? Oh, sorry, this was at the time they were unable to sequence regions of it. They knew about how long it was based on their methodology, but they didn't know what that sequence was. So this was the sequence they published. Since then more complete sequences have been published. Okay, but there are different formats to present DNA data. This format here, where we have 10 base pairs and then 10 more and then 10 more and numbers along the side, is called Genbank Format. And so this is the one that is commonly used when you're depositing your sequences in Genbank. It's a little bit of a complex format, but it allows for all of this information also. So in reality, we start up here. All of this is Genbank Format. And this is a really good format that is data rich. You can put lots and lots of information in it with all these keywords and everything. For many applications, simpler formats are a little bit better. So I'm going to talk about two formats that are in the lab that you just need to be a little bit familiar with. Okay, the first one is Fasta Format. Fasta Format is the simplest format. So I'm going to click on this. It's going to take a minute to load here. The Fasta comes from, some people call it Fasta. I've heard it pronounced both ways. I'm having a little trouble, so while that loads, let's look at the one that's in the lab. Okay, so this is Fasta or Fasta format. Okay, and the way it works is it's very, very simple. It's simply a text document. So you can look at this in like Note. It's called Notepad on PCs. I forget what it's called on Macs, but any text editor, you can look at it in Word if you want to. So on the first line, we've got this little caret signal or greater than, right? So that little signal designates that this line is going to be the beginning of the first sequence. And then a sequence name. And you can make this, as long as you don't have another line, you can make this as long as you want. So you can put descriptions in here, you can put extra information, whatever you want in the sequence name. Then there is a return, so on the next line, and the sequence. Now I put the dots here just to represent that it can be longer, but A, T, C's, and G's. You can also have N's in the sequence, right, which represents, I know there's a base pair there, but I don't know which of the 4 base pairs it is. Okay, but if everything's been sequenced thoroughly, then you'll just see A's, T's, C's, and G's representing the sequence. And then that's repeated for sequence 2, sequence 3, sequence 4, and so on. So it's a very, very simple sequence. And so we will be interacting with sequences a little bit, but you don't need to worry a ton about it. But I just want to introduce this because it's a common, very simple format. And it is the basis of our next one, which will be a little bit important as we move on in future labs as we are assembling a transcriptome. Okay, so Fastq format is very similar. Let's go back and see if we can look at the Fasta format for, there it is. So here it is, simply put, here is the format. Okay, so we've got a description. This one has the accession number, which is that unique number assigned every sequence, and then a description of what it was when these people published it, and then the sequence itself. Now here you've got carriage returns, but in reality, if you download this sequence, this all will be on one line. So this entire sequence here will all be on one line. Okay, so that is a simple sequence format called Fasta. Fastq is just a little bit different. It's based on Fasta, so starts the same way, where we have sequence 1, however, whatever description you want to give it, and then we have the sequence. Line number 3 is simply a plus that represents a separation between the name, the sequence, and then what comes next. And what comes next is a series of characters. On this line, you can use all, I think there are 94 of them, but all of the basic characters on a keyboard, all the computer characters. It's ASCII, I forget the name of it. But anyway, so each of these represents a quality of the base pair. So this first one corresponds with that first base pair, the A. Okay, and so that represents a certain quality score, 1 through 94 basically. This would be a very low. And it uses all of the symbols, all the capital letters, all the lower levels, so there are 94 different symbols, and they're ranked from lowest to highest. So the key idea here, and then it goes on, so then you can put sequence 2 and sequence 3 with 4 lines for each sequence. The key idea here is not only do we know the sequence, but we have an estimate for how much we trust what the quality level of that sequence is. And so this is what we use when we use raw sequencing data. And so that way, if I have 2 sequences that overlap with different quality scores, I can make an assessment about which one I'm going to trust. And hopefully we've got 3, 4, or 5. And if we have one that disagrees, that's at very low quality, and 3 or 4 that are very high quality, we can be very, very confident that we have sequenced that our data is correct. Because there are some errors, right? It's like any observation you make in the field, depending on the methodologies you're using to make that observation, you might have various levels of certainty in the quality of that observation. And so Fastq is simply a format that gives you the sequence and an estimate of the quality of that sequence, and is used when we are doing DNA sequencing for genomes or for genes or for transcriptomes. Okay, so that's the answer to number 3. Now the last part that I want to show you is a BLAST search. Okay, a BLAST, and BLAST is another fun acronym. You get lots of acronyms, well, really with any technical field. My wife is a nurse, and I almost had to learn a new language so I can talk with her about what she does on a daily basis because they use so many acronyms. So would come home. Some of you going to the medical field will appreciate this. She came home and said, "Hey, we did a cabbage today." This was when she was working in the OR. And I was pretty certain she wasn't referring to vegetables, right? But I said, "Okay, tell me what a cabbage is." And she says, "Oh, coronary bypass with arterial graft." Right? It's basically a heart bypass, but there they call it a cabbage. So anyway, BLAST is also an algorithm. So let's look at this. So here is a little thing that allows us to run BLAST. And we can run BLAST on this sequence. It will automatically load this sequence if I'm on a page with the sequence here. Okay, so BLAST is essentially a search algorithm that allows you to find close similar matches in the database. Okay. While I'm broadcasting, while I'm recording, I use up a lot of my bandwidth here, which is why my internet runs so slow while I'm also recording a lecture here. So we'll let this run for a little bit. If it doesn't come up, that's fine. We're going to be actually interacting with BLAST a little bit later on. Oh, here it is. Okay. So you can put raw sequence in here, or you can put the accession number, and then it will just use that Genbank accession number to BLAST it. Okay. You can also, if you want, insert a job title. There are lots of other things you can do. We're not going to worry too much about this yet, but there is one thing that I would like to recognize, that you can limit the search. So if I search for this now, it's going to search for all of the DNA sequences that match or are similar to this opsin in the fruit fly. But let's say I wanted to speed things up a little bit and I didn't care about finding matches in jellyfish or matches in vertebrates. Instead, I would just like to find insect matches. So I'm going to say, oh, let's look for insects. Here we go. I'm going to click on that option, and now it's limiting it. Here's that tax identifier we talked about earlier. It's going to limit it to only insects. So now when I search, it's going to find all of the closest matches to this gene among the insects. So again, you can think of it like a Google search, but for a gene and just on this database. But it's a massive, massive database. And so it will take a minute or two for this search to complete. But when it does, here we go, you will see a results of your search. And so notice, of course, the very first thing it found was a Drosophila melanogaster opsin gene, 100% identical. Okay, now it's finding other genes in other melanogaster strains. So this gene has been published multiple times because there's so much work on melanogaster. It's found chromosomes and pieces of chromosomes. And then as we move down a little bit, now we're going to start to see matches that are outside of Drosophila. So here's a match for another opsin gene, but in a different species of Drosophila, and then other species. And if we go down, because there's so much Drosophila sequence, we're probably not going to see anything outside of the genus. But if this was a different organism, or if we had limited the search differently, we would begin to see other more divergent. But here we're finding only Drosophila ones. Okay, so it's a powerful search. A BLAST search allows you to identify what a sequence is by looking at similar sequences, and then also find homologous genes just based purely on sequence similarity. One of the challenges of this lab, it's new, and so basically you guys are my guinea pigs, but also it's an online format, which is very unusual for biology labs. And there are going to be some difficulties, some technical difficulties. You might come up with problems. Please don't freak out if you're having troubles. Contact me. Don't spend a ton of time troubleshooting. If you think maybe you can figure it out, keep going. But if you're having problems, let me know. I'm going to try and limit the amount of technical glitches, but again, because it's the first time, I have some idea that they're going to be some, but I can't anticipate all of them. So I'm going to have you download a piece of software. It's available for both Windows and Macintosh. Much of what we do from now on will be web-based, which hopefully will streamline it a little bit, make it a little bit easier. But occasionally, maybe one other time, there's going to be some software that we need to use. And again, if you're having troubles with it, let me know. Okay, so I want you to go to megasoftware.net. Mega is a program that is freely available. It was generated under a grant, and it's very useful. It can do all sorts of analyses. We're not going to use it for all of its purposes. You may even find it useful if you're working in a lab or, I don't know, for a higher level biology course. But there are many other programs. Some are pay for and are very expensive, but this one will do what we need, and we're only going to use it a little bit. So go to megasoftware.net, choose your version, either Windows or Mac. Linux are available also, but I don't think you'll be any Linux users in this class. And then download it. And go ahead and install that on your computer. And then if you want, you can pause the video and follow along. So when you open Mega, you have an initial interface window. One of the things that's a little bit confusing for some new users is that Mega uses multiple windows. So don't be confused, just realize there's going to be more than one window open for Mega. Okay? I have provided a data file for you of Drosophila opsin sequences. Drosophila has 7 identified opsin sequences, and I have made a data set that is available on the lab page. I want you to download it and open it. So once you've got it downloaded and Mega installed, you're going to open Mega and go File, Open a File Session, and then simply navigate to where it's located. So it's going to be called Drosophila opsins, and you're going to want to choose the Align version. So this is just all 7 DNA sequences in their raw format. And let's just take a look at it. A's, T's, C's, and G's. I have the names here. This first one is RH1, RH2, RH3, and then so on down the line. And you should be able to see the same thing when you open this file in Mega. Now, one of the nice things about Mega is you can look at it at the DNA view, or you can look at it at the protein level. So when I click this little tab up here, Translate Protein Sequences, I'm going to get a dialog message asking which genetic code I want to use. There's slight differences for mitochondrial versus nuclear, for prokaryotic versus eukaryotic. We're just going to use the standard eukaryotic one. You can click that little arrow so the dialog box doesn't come up anymore, and then say yes. Right? So here we go. So now each of these letters represents an amino acid. Notice they all start with methionine. That's the start codon. And there are methionines in other intervals, other places. So methionine can be inside of a gene also. Now notice that we get really, really divergent. And if you scan down, you might notice some patterns, but it's kind of a mess. And the reason is because of insertion-deletion mutations, we don't have a good alignment. And if we look at the DNA view, you're going to maybe even see it worse, where it's just maybe a few again. As you scan down, you see a few patterns, but not much going on because they are out of alignment. So the first thing that I want you to do is go back to the protein view. So make sure you're on protein view. And then we are going to perform a quick alignment of this data set. And there's algorithms or software here to do that. Okay, so what I want you to do is you can either do Control A, which will select all of them, or an alternative, you can click on the first one, hold down the Shift key, and click on the last one, and that will select all of the sequences. 2 algorithm alignments, we're not going to worry about how this works. It's a topic for another class, but we've got one called Clustal, which is this little W icon, and one called Muscle, which is this little bicep. You can use Muscle. Either one really probably give you a very similar alignment. So we're going to click on that and say Align Proteins. Leave everything at the defaults and click OK. So it's going to do a quick jump through and align this. Now, here's something that you are going to notice. There are some areas that now we can see very clear patterns. Although there are mutations and changes, it's very clear that those are similar sections. And then there are other sections where it's maybe a little less. It's a little crazier, right? Particularly at the beginning here, the N terminus. And I've got a question on the worksheet, so let's go ahead and answer that one. And we're actually looking at the data to answer this. So compare the diagram of Opsin domains, which we've got up here. All right, here's our diagram of Opsin domains to my alignment. And then let's see, of the 7 opsins found in Drosophila, based on your observations, which of the domains are under the least constraint? So let's talk about what that means. Constraint is an evolutionary term. It basically means that if mutations occur, they're very rarely going to be kept, particularly at the amino acid level, because that part of the protein has a very specific important job to do, and most mutations are not going to be beneficial. So constraint means that we're going to see similarities between all of the sequences in our alignment. Okay, so one issue that we've got here is that we don't know where the domains are. Okay, so to help you through this. Now what you could do is we could compare the diagram and count out exactly how many amino acid residues and find out all of the domains. I've already done that for you. You don't have access to this file, so I'm going to come back to the one that you have here. You've done the alignment. You can just leave it open and watch for now, and then I will come back to this aligned file. So I'm going to go ahead and save this version. And I'm going to open a different version here. Again, you don't have this one, that's fine. And this is one where I have already separated the sequences based on that domain. So I'm going to translate it into amino acids. And here's what I have. So this first region, all as messy as it is, represents that N-terminus. And then I've put a group of spaces to represent our transition to the next domain. So this is our first transmembrane domain. Then we've got a space. We have our cytoplasmic loop, the first one, and then the next transmembrane domain. So if you refer back to this diagram, that's basically what we're looking at in sequence formation, in sequence form. The N-terminus, transmembrane domain. Cytoplasmic loop, transmembrane domain. The extracellular loop, and then the next transmembrane domain. So they're separated out. So this will help us to answer this question. So the question was, which of these domains has the most constraint, meaning that they are the most similar? And I don't expect, a detailed analysis here. But notice that there are some sequences that seem to be very closely related to one another and have changed a little bit, but are still recognizable as part of the alignment. So the N-terminus appears to have the least amount of constraint, at least of the ones we've looked at so far, right? Look at all that variation. Although some sequences are similar, even between sequences, there's lots and lots of variation. So N-terminus does not have much constraint. Look at the transmembrane domains, quite similar. Transmembrane domain 1, transmembrane domain 2, and you'll notice they're very similar in length also. Transmembrane domain 3, and then so on. So transmembrane domains are much more conserved. They have more constraint. And then let's go all the way down to the C-terminus, and notice again that only a little less, especially at the very end of it. There's some conservation there. So the transmembrane domains, and to some extent, not all, because this is a one of those loops, there's not a ton of constraint there in the loop. Again, some variation here in length. One of these loops is very different size than the others, with some constraint in the others. And so this can be a hint as to whether that has a conserved function. Okay, so to answer this question, number 5, right? Based on our observations, which of the domains are under the least constraint? Probably the best answer would be the C-terminus and the N-terminus. Those are the ones that seem to have evolved and vary the most, okay? And then to follow up, which of these 7 opsins is the most divergent? So let's look at that. I'm going to give this one to you because this can be a little tricky. So let's just look at all of the 7 opsins. So notice that we see clear patterns of alignment between 1 through 6, even though there's some divergence here. Alignment, right? But notice that this one on the bottom, which is Drosophila melanogaster R7 or Rhodopsin 7, is quite different from all of the others, right? We've got that one's quite a different sequence, even though it's conserved across many of these others. We have another good example here where we've got a big deletion, a gap. We're missing part of this loop for that RH7, and then as we get to the end, it's much longer, much different from all of the others. So this RH7 is the most divergent, the most different of all of these opsin genes. Okay, now the hint says for you to Google it. So let's look up Rhodopsin 7. So I'm going to come up here and I'm going to say Drosophila. We'll just do the abbreviation. That should be good enough. RH7. And see what comes up. Okay, so our first link is a paper that looks at this different gene. And if we scan down, we have this interesting one. It says the unusual rhodopsin in Drosophila. So let's take a look at that one. It's a document at Ncbi. Pretty trustworthy. Okay, so here we are. We'll just scan through the abstract. We don't need all of the details. Rhodopsins are the major photopigments of flies. Drosophila has 6 well-expressed rhodopsins, which are the 1 through 6, 7, and there's an R7. It's highly conserved, right? But phylogenetic trees cluster in 3 different groups. All those form one group, all those form another, and RH7 forms its own. So RH7 is unique and probably has a distinct function, and we might have even guessed that just based on the sequence differences. Okay, so RH7 is a very different from these other 6. Here's their absorption spectrums, kind of talks about where they're expressed in the eye. So RH7 has potentially quite a different function. Now, one of the things that we've talked about is there are some opsins that have reverted back to a different function that might be functioning in the brain, might still be interacting with light, but in a different way, or may be even in a light an environment without light, and so might have gone back to a protein. So this would be a good candidate for exploring why is it different. So these are the sorts of things that we can do when we can analyze the sequences of organisms. Now, to finish, I've got one last assignment for you in Mega. So I'm going to go back to our original alignment, the one that you just kind of paused on, and I'm going to help walk you through this last component of this assignment. Okay, so this should be where we left you off. You've got all of your Drosophila sequences, which you downloaded, and now they are aligned. So if we look at them in the nucleic acid view, you're going to see regions that are very similar. The domains are not separated out in this version, but you're going to see a lot of conservation. And again, as you scan down now, you can see R7, right? The bottom one. Notice how different it is from the others as we scan across. It has large gaps where none of the others do. And especially at the ends, it's quite different. Okay, so again, signaling probably a very divergent function for that gene. Now, the last assignment, and this is just to kind of show you some of the options, is I would like you to add one additional opsin sequence to this and then align them, and then you'll save that file and turn it in as part of your lab. So you can choose whatever organism you want. If you want to target the organism that you were assigned for the previous labs or one of those organisms, that's great. I will do a demonstration. You can do this one if you'd like. I just like you to kind of get a feel for some of the options, and we're not going to use this program a lot, but it's useful. Okay, so there are 2 ways that you can go about this. So let me show you both. And there is a web browser and a simple way to add sequences to your Mega alignment. So at the top here, you have 2 little shortcuts. This one is a blast search, which we talked about previously. I'll show you that. And I'll add a sequence using both methods. You can do either one. This one is simply opens a web browser to Genbank. So once it opens, you will see this. And I'm going to change it to nucleotide. There we go. And then I'm going to search for an opsin. Now, when I do this, it's searching for opsins across all of the database. But let's say I want one for honeybee. So I'll put in keywords, and keyword searches sometimes don't work with this, but we'll give it a shot and see. So here we have Apis mellifera, a long wavelength opsin. Great. I kind of took a chance, but it came up. So let's go ahead and add that one. And if you'd like to use a different sequence, that's fine. So here we go. Now, one of the nice things about the Mega web browser is it has this little button, Add to Alignment. So when you have a gene that you would like to add, rather than cutting and pasting it and making a new sequence, you can just simply click this little Add to Alignment. The default is to import only the coding sequence, which is great. And we're going to say OK. You can change the name if you want or just leave the default. So I'm going to say OK. So there we go. So now I have an additional sequence added to this already done alignment. So once you have your sequence added, if you want to use the B, that's fine. If you're interested and want to play around, pick a different organism. Do the one that you were assigned in the previous lab. So to do an alignment, because I brought this in as an unaligned sequence, so if I look at this bottom sequence now, it's not going to match up very well with any of the others. So I want to align it. So same thing, all I'm going to do is use Control A. You want to do this in the protein view because proteins are going to be a little bit more conserved than the nucleic acids. So Control A, or pick the top one, hold down Shift, click the bottom one to select all the sequences. And then let's do a new alignment. So I'm going to do Mega, Align Protein, and hit OK. So now my bee sequence is lined up with the fruit fly sequences. And I could potentially even compare it to say, oh, is it more similar to some of them than to the others? Maybe I could find a match, a closer match amongst some than the others. So you're starting to get an idea of what you can do, how powerful this type of analysis may be. Okay, so just be aware, it's kind of a fun thing to do. Once you've got it done, if you want to play around a little bit, that's fine. I'm going to show you one other way that you can bring in a sequence, and then I'll let you finish up your alignment, save your alignment, and this will be one of the things you need to turn in for this lab. So you're going to have three, well, basically just two documents that you need to turn in with this lab. You need to turn in the lab questions. You need to turn in your alignment with your added species, okay, and just turn that in. And then make sure you answer the lab questions. That's part of the lab, but you can answer those, and then there will be like kind of a reading quiz type thing, but a lab quiz for you to also put your answers for some of those questions. So complete the lab completely, and then do those lab questions. That will be the easiest way to do it. So one other way that you can do it. So I'm going to go ahead and delete. Excuse me. I'm going to go ahead and delete this sequence and show you a different way to do it. So let's say that we wanted to bring in a sequence that was most similar to this unusual RH7. And maybe it's available in other species, maybe not. But let's say we want to do that. So what I'm going to do, it's probably easiest to go from the nucleotide view. I'm going to click on this one, and then I'm going to perform a blast search. So the link is right here. And if you've got a sequence highlighted, it will automatically load that sequence into the blast search. Okay, my blast search is having technical difficulties because I'm trying to record a video and upload it while I'm accessing the internet. So it's not that important. Don't worry about, just know that you can add sequences by blast searching, finding similar sequences and adding them that way. And that's really all you need to know. Okay, so once you've got your sequence added, just use the taxonomy search option that I showed you. Once you've got it added, do an alignment, and then go ahead and save that. So you'll just hit save. You can save it under whatever name you want. And then you're going to add it as an attachment to the lab assignment for the completion of that part. The last part of Lab 5 is very short and simple, but also very important because you need an account on this server web platform so that you can complete the remaining labs. So there's a link in the lab. If you'd like to do that, just click on that, or you can look at Cyverse Discovery Environment and Google that. So you need to go to this page. There is a sign up thing here, your name, last name. You can make whatever username you want. You can use your university email here if you want. And then just complete, I'll just show you an example. I already have an account, so we'll just make a new one using my personal email. There's my personal email if you guys want to send me a message. No, not a good idea. Send me to the university email. Oh, it looks like I've already done one. So let's just I've got another. Let's just put in a random one. I think it will ask you to confirm the email. So make sure you have a workable email. You can use your university one. And then you can just put your, UTRGV, biology, enter student, where are we? Undergrad student, research area, bioinformatics is fine. It's essentially what we're doing, a version of that. And you can say not applicable because we don't have a funding agency for this class, okay? Maybe University of Texas. Where are we? Do we not have it on here? Well, we can enter just UT Austin if we need to, but it's probably here somewhere. Am I just missing it? Oh, there it is. Okay, for some reason I just didn't see it there. Okay. Okay, and again, so just go ahead and fill this all out. Now, once you have everything identified, you'll be added to the system. Go through it. There. This is just because this is supplied by grants, so you're going to want to know United States, United. There we go. There we go. Again, they just are asking for this information for their grants, and so they can keep track of who's using their material. So again, whatever you feel confident or comfortable with, Texas, ethnicity, again, whichever one you want to do. which applies best to you. If you can also do not applicable or you choose not to do it. And then this is instructor, right? So go through that, go ahead and submit that, and then go to your email, confirm it, and then you'll activate your account. Once you have an activated account, send me your username. I'm going to share data with you for labs 6 through however many we end up doing for the semester. All right, that's it. Finish lab 2 or lab 5. If you have any questions, please contact me. I'm going to allow extra time for this. I had some technical difficulties in getting it up. I'm going to work over the break to get caught up also, so that our previous labs will have a whole week and ample time, because we are going to be facing probably a couple of little unanticipated issues. There might be some user issues.